Hi, and welcome back. This is part 3 of the 3-part lecture for chapter 5, cell division. The first part covered um, chromosomes and chromosome structure and organization. Part 2 covered mitosis, and part 3 here will cover meiosis. Before we get into meiosis, let's understand the different kinds of cells in multicellular organisms. There's two basic kinds of cells. There's body cells, which are called somatic cells. Remember that soma is the root for body. And somatic cells are everywhere. Most of your cells, almost all of your cells, are somatic cells. And somatic cells divide by mitosis, right? Mitotic cell division allows somatic cells to reproduce. This is your skin, your heart, your liver, your kidneys, whatever. Everything in your body is made of somatic cells. And then in one specialized organ, you make sex cells. Okay, and so sex cells are specialized cells that bodies produce that are only used for reproduction. And the way sex cells are created is through a special kind of cell division we call meiosis, or meiotic cell division. Okay, and remember that both of these, meiosis and mitosis, cells going through either one of those still progress from G1 to S to G2. Okay, and it's just the M that's different. Okay, so here we go. In meiosis, as we said, it occurs in the life cycle only of sexually reproducing organisms, and the goal of meiosis is to create 4 cells instead of 2 cells, 4 cells that are as genetically different from each other as possible. Okay, and so each of these cells has only one set of chromosomes. So in humans, these cells will each have 23 chromosomes instead of 46, right? So remember that the N of humans is equal to 23, and the 2N or diploid number is 46, and a somatic cell has 46 chromosomes in its nucleus. In a sex cell, it will only have 23 in here. Okay, so you would say that sex cells are haploid, whereas somatic cells are diploid. Okay, these words are used to describe the number of copies of each given chromosome. Okay, so you can also think of all the chromosomes as a complement of chromosomes, or a set of chromosomes. So how many sets of chromosomes does a cell have? Okay, and when I say set of chromosomes, think of a set of socks, right? So if you just have one unpaired smelly sock, that one sock, okay, versus having the other pair, the other sock, okay, having 2 pairs of socks. So if you have 2 pairs of socks, that would be 2N or diploid. And if you only have one sock, these are terrible socks, that would just be N or haploid. Okay? Okay, so the goal of meiosis is to take one cell, that is 2N, and to reduce that into 4 cells, 2, 3, 4, that are N. Okay, and the way we do that, very simply, is to divide the cell first in half. So go through a division where we reduce that number of chromosomes from 2N into N. Okay, and then each of these in turn divides into 2 daughter cells that are also N. Okay, and we'll learn N. We'll learn that what happens here is, again, it goes back to those sisters. Okay, so the sisters here is where they're going to separate. Okay, in the 2N cell going to the N cell, what's going to happen is the homologs are going to separate. So the pair of homologs separates. And if this is confusing, it's because I haven't shown you the rest of the slides. So that's just a very brief overview. Here we go. Okay, so again, we begin with one diploid parent cell. Okay, so if this is an example cell, this cell's nucleus will have, let's say, 1 chromosome, 2 chromosomes. Okay? And we're going to take that and we're going to reduce the chromosomes until the cells look like this. And yeah, that it'll just look like that. Okay, so it starts with 1 diploid cell. It ends with 4 haploid cells. Okay. And the key here is that during the first during the first stage of meiosis, the homologs line up side by side. Okay, so here we have 1 chromosome, that's say dad's chromosome 1, and mom's chromosome 1 sits right next to his. Okay, that's the key difference between mitosis and meiosis. In mitosis, okay, those would simply line up like this in any order, so that when they get yanked apart during anaphase, each one of these sisters goes its own way, and the resulting cells each have one of those sisters, right? Each cell has one of those sisters. In meiosis, okay, the first cell division reduces the number of copies each chromosome. Okay, and so this cell will have 1 chromosome, and this cell will have 1 chromosome. Notice they still have 2 sisters, right? And then eventually these will separate, and each one of those cells will have one of those sisters. Okay? Okay. Now the key here, one of the keys here, is that DNA replication doesn't occur between meiosis 1 and meiosis 2. These 2 cell divisions happen consecutively, quickly, right next to each other. Okay. Okay, and I'll tell you what, rather than just read these slides, I'm going to skip ahead to the diagrams and really just explain it to you. You can use these PowerPoints to study on your own as well. Okay, so here we go. This is a cell that is going through meiosis. Okay, this cell right here. This cell is 2N, and you can see the 2N number is 4, which means 1, 2, 3. There's 4 chromosomes here. Okay, there's, notice, a short one, this little short one here that's red, and the corresponding homolog is this short blue one, and then there's a long chromosome that's blue and a long chromosome that's red. Okay, so the N number is 2, right? There's a long chromosome and there's a short chromosome. During the first meiosis, you can see the cell goes from 1 cell with an N of 4 to 2 cells, each with an N of 2. Okay, so we've reduced the number of chromosomes. Okay, the original cell had 1 of each, so right, think of 2 pairs of socks. Okay, and what we did was we basically took each of those pairs and separated them. Okay, and then once the homologs are separated, we do one more division and we separate those sisters, right? So that each cell that results from this division is now haploid and has only one copy of each chromosome. Okay, so fertilization is simply when we have one gamete, the egg in this case, gets fertilized by the sperm, and so what really happens is the DNA from the sperm gets injected into here, and so the sperm, which is N, mixes with the egg's nucleus, which has N, and so that yields the zygote, which is 2N. Okay. All right, so let's go into a little more detail here now. Meiosis 1, just like mitosis, has 4 phases. There's prophase. These are out of order here. So we'll correct that later on. For now, understand that these are inverted, and I'm not sure why. Okay, so it's prophase, and it's prophase Roman numeral 1 to signify the first meiotic division. So prophase 1, metaphase 1, anaphase 1, telophase 1. Okay, and I skipped here prometaphase. This slide for some reason doesn't have prometaphase, which I'm just going to scribble here because I know that you're just sitting here watching me right here. Prometaphase 1. Okay, and so meiosis 1 has prophase, metaphase, anaphase, telophase, or prophase, prometaphase, metaphase, anaphase, telophase. Okay, and during the first division, there are some fancy things that happen that ensure genetic variation occurs. Okay, so we're going to learn about two of these mechanisms. One is called crossing over, and the other is called independent assortment. All right, so here's the first division. During the first division of meiosis, okay, just like in mitosis, you can see the chromosomes condense. Now notice here, the chromosomes are intermingled with each other. This is where something called crossing over occurs. Okay, crossing over is when two non-sister chromatids exchange chunks of each other, as you can see here. You can see that in the small chromosome, a little bit of the tip of this guy here was exchanged with that guy there. Okay, and in the big chromosome, you can see a little bit of the bottom was exchanged. Okay, the way this occurs is during prophase 1, the homologs, okay, so here is one chromosome, and here would be, let me change the color of this pen here so you can get a better idea. Let's do red. All right, so there's chromosome 1 from mom. Okay, so mom and dad's chromosomes, and what happens is these areas basically switch a little bit of information. The way they do this is first they line up, they pair up, and this process is called synapsis. Okay, so synapsis is the process by which the homologs pair up. Okay, and then after synapsis happens, then you have the crossing over. Okay, the crossing over. Okay, so all that happens in meiosis 1. Prophase 1 of meiosis, crossing over occurs. Okay, that's the key to meiosis 1's prophase 1. The next key difference is in metaphase 1. Okay, notice in metaphase 1, the homologs are side by side. They pair up. Okay, so there is organization here. In mitosis, the difference here would be, if this was mitosis, each one of those 4 chromosomes, would be lined up on the metaphase plate like this, okay, as opposed to what you're seeing here, which is they actually pair up side by side like this, okay? So this would be mitosis, where they just all randomly line up, and the reason it's random is because it doesn't matter. Both sides are getting exactly the same thing, okay? In meiosis, okay, this homologous pair lining up means that one of these homologs is going to go this way, and the other one is going to go that way. Okay, and remember, these chromosomes are different from each other. They have different alleles. Even though they have the same genes, they're different. Remember, these would be the genes that carry, say, the green eye color versus the blue eye color. Okay, so when these separate, when these two chromosomes go into this cell here, and this cell now has only 2 chromosomes instead of 4, okay, this now has less information than the original cell. This chromosome, okay, versus this chromosome are different, and now they're in different cells. So this cell has a distinct genome compared to this cell, okay? And what's really going on here is for every single one of those copies of chromosomes, every single one of those pairs of homologs, okay? So let's say here is chromosome 1, and there's dad's copy, and here's mom's copy, okay? And here's chromosome 2. Here's mom's copy, and here's dad's copy. And here's chromosome 3. Let's say it's dad on this side and mom on this side. Okay, and chromosome 4, let's say mom's is on this side and dad's is on this side. So that this pairing up is random. Okay, and each set of homologs pairs randomly on left or to the left or to the right compared to each other. There's no connection between any of these guys. So that's independently assorting. So that's what this is called. This is called independent assortment. Okay? When the homologs randomly segregate to either one of the daughter cells. So you can see that action here. Okay? These two, in this case, in this scenario, this chromosome went left and this chromosome went right. However, in another scenario, this could have been over here instead and went this way. Okay, so this cell could have gotten either this blue one or this red one. It's random. Okay, so you can see here that this blue one and this red one make up this cell. But you can imagine another scenario where this small one was also red and this was red. You can also imagine a scenario where this blue one is here and this is blue instead. Okay, so there's all the different possible combinations, and that's what gives rise in one way to the genetic diversity of the offspring of the the resulting cells. Okay, and then the second way is through that crossing over. Okay, so remember this chromosome here, now that it has that little chunk of the other chromosome, that's a new chromosome. There's new combinations of alleles now that have never been seen before. So between this shuffling of the chromosomes, between the shuffling of the homologous pairs, okay, between this shuffling, this crossing over, and the fact that these are going to segregate independently from each other, the result is 4 cells that are completely genetically distinct. Okay, so here we are at the end of meiosis 1, and you can see that these 2 cells aren't done yet, right? We still have to go and divide further. What else can I say about meiosis 1? So after anaphase 1, after the homologs separate, that's what you would say here, the homologs separate. Okay, then the nuclear envelope reforms, and the cells go through, instead of cytokinesis, we call this interkinesis. Okay, during interkinesis, the nucleus basically reforms only to start fragmenting again when we hit meiosis 2. Okay, here's just a zoom in for the first division for your own studying. Okay. All right, now these slides just reiterate everything we just talked about, so I'm going to skim over this and you can read these on your own. This is a good example of how crossing over works. Okay, so let's clarify what we're seeing here. This is a duplicated chromosome. Okay, you can see 2 sister chromatids, 1 and 2. All right, you can see the kinetochores on the centromeres. Okay, and then this is mom's chromosome. Mom's chromosome. Okay, so notice that they're paired up next to each other. This is what happens during prophase 1. This is synapsis. Okay, there's that word, synapsis of homologs. They pair up, they find each other. Okay, they're all paired up perfectly. And then what happens is, in reality, this happens about 7 times in random, completely random places along every single chromosome. What happens is one of these arms of the sister chromatid on this side twists around this sister. So the relationship between this chromatid and this chromatid, these are non-sister chromatids, non-sister chromatids. And it makes sense. Okay, it makes sense because if crossing over occurred between sister chromatids, then we wouldn't have any, there would still be the same exact sequences. That doesn't make any sense, right? So if non-sisters cross over, and you can see here, there's the crossing over, okay? And then what happens is there's enzymes that basically cut and paste these guys so that they basically exchange pieces. So this brand new sequence here, these are completely new chromosomes. Nobody's ever seen these combinations before. Okay, so this chromosome here, okay, rather this chromatid here, that's the original chromatid, right? And when they separate during meiosis 2, which we haven't gotten to yet, but you can see here, this is a standalone chromosome now, right? Notice that it is just like dad's, and this one here is just like mom's, okay? So we call the original ones the parental chromosomes, because they are the original parent sequences, okay? Just like mom used to make, just like dad used to make. And then these new ones in the middle here, we call them recombinant chromosomes because they're a combination of the original shuffling, right? So you can see this pair here we call the recombinant chromosomes. Here's the word over here, recombinant chromosomes, okay? So there's the parental chromosomes and there's the recombinant chromosomes, okay? And in real life, this occurs about 7 times. Some pictures that you see of this might have letters associated with the different alleles. So for example, there might be A, B, C, D, E. Okay, and these just correspond to regions of genes. Okay, so let's just pretend these are all capital letters here. D, E, okay, and then these are lowercase letters. C, D, E, A, B, C, D, E. Okay, so when we have this crossing over, long story short, these will still have the original alleles all in the same order. But these, watch this, capital A, capital B, capital C, capital D, lowercase e. Okay, lowercase a, lowercase b, lowercase c, lowercase d, capital E. Okay, so you can see here this capital D being next to a lowercase e for the very first time. Those two genes have never seen each other before until now. This would be the equivalent of, say, a red hair gene here and a freckle gene here. Okay, so normally, rather, red hair and freckles. Okay, and this would be brown hair and smooth skin. And so this would be red hair and smooth skin, and this would be brown hair and freckles. Okay, so that's why it's so rare to see people with red hair with fair skin and people with darker hair with freckles, because they're very close together. We would call those two genes linked because they're very close together. Okay, so metaphase 1. After that crossing over occurs, the homologs pair up at the metaphase plate. Okay, and it is completely random which side the maternal and paternal members are oriented. Okay, when we say toward either pole, the pole they're talking about is the centrosomes. Okay, so when we have the spindle formed like this, these are the poles that they're talking about in this slide. Okay, and so mom's is over here and dad's is over here, or it's the other way around. Okay. Okay, so this is a really good example of how independent assortment works. On the left, you can see that there's the blue short chromosome next to the red short chromosome. Okay, so dad's is on the left and mom's is on the right. For the big chromosome, dad's is on the left, mom's is on the right. Okay, so the two blue ones are on the same pole, and these two red ones are on the same pole. And so as a result, when anaphase 1 occurs and these homologs separate, the resulting cells, in this case, in this example, both of these cells each have all of dad and all of mom. Okay, that's one scenario. But it's also just as likely that instead of the blue one and the red one and the blue one and the red one, like on the other side here, we have the blue one and the red one, but now the red big one is on the left. Okay, so basically these guys swapped. Okay, see? So now the blue one is on the right and the red one is now on the left. And so as a result, the small blue one and the big red one go into this cell. and the small red one and the big blue one go into this cell. And so as a result, these two cells are completely different, right, from these two cells. And if you look closely now overall, this cell is different than this cell is different than this cell is different than this cell. 4 different cells. Increased genetic diversity, and therefore a greater chance of survival in a in a changing environment. Okay, and this depicts a really good crossing over. So you can see synapsis occurs. Synapsis is when the homologs pair up. Okay, and they exchange pieces, and then during metaphase 1 you can see the results of those crossing over. Okay, also notice those are 4 completely new chromosomes compared to the original. Okay, so so think about independent assortment too. So you have the chances of getting this guy and this guy, right? Or you can get this guy and this guy, or you can get this guy and this guy. Okay, you can think of all the different possible combinations here. Lots and lots of diversity. Okay, this is just a restating of everything we just talked about. All right, so here, really hits home how much variation there is. So in humans, because we have 23 pairs of chromosomes, the number of possible combinations is 2 to the 23rd, or over 8 million. Okay, now remember, this value doesn't include genetic recombination. It doesn't include what we call crossing over. Okay, when they say genetic recombination, that's just a general term we use to describe mixing and matching DNA. So crossing over is an example of genetic recombination. All right, so for each one of these 8,388,608 different combinations of 23 chromosomes, for each given set of chromosomes, 7 times over, and each one of those 23 chromosomes, different pieces exchange from crossing over. Okay, so you can imagine just for a single chromosome, okay, where there's thousands of genes here, and exchanging huge chunks 7 times, essentially the possible combinations of the human genome are almost infinite. Almost. It's a number that's bigger than the universe itself. All right, telophase 1 sometimes happens, sometimes doesn't. The nuclear envelope reforms, the nucleoli reappear, cytokinesis may occur, producing 2 daughter cells which are haploid. Notice that there's a lot of mays and maybes here. That's because during specific kinds of meiosis, this doesn't happen. We'll learn when we look at oogenesis, which is the formation of the egg cells in humans, or in mammals. All right, so again, here's anaphase 1 and telophase 1. You can see in this case the homologs separating. And this is where independent assortment really cashes in. Okay. And there's interkinesis, which is just that period of time between the first and second meioses. Okay, and now we talk about the second cell division, which again is prophase, metaphase, anaphase, telophase, except with a Roman numeral 2. And essentially what I'll say about this is that it is pretty much the same as mitosis. The only difference being that there's only 1 set of chromosomes, okay, instead of 2 sets of chromosomes. During prophase 2, the cells each have 1 chromosome from each homologous pair. Remember, these are duplicated chromosomes though, right? They look like this still. They're sisters, okay, except there's just not the other homolog anymore. So spindles appear, the nuclear envelope disassembles, each duplicated chromatid attaches to the spindle. Again, this is all mitosis, so everything you learned in mitosis, it's exactly the same. Okay, there's a metaphase plate where they all line up in the middle. There's just half the amount now. During anaphase, the sisters separate. Okay, so we have that separation of the sisters that happens. And then in telophase 2, the spindle disappears, the nuclear envelope reforms, and we get cytokinesis. Okay, So here's meiosis 2, and notice that it's happening twice, right? This and this that happens in duplicate, these are just exactly the same. Notice though, you can see the little details here, right? Because of this crossing over that occurred, and because of the independent assortment that forced the small blue one on this side and the small red one on this side, we could just as easily make this entire slide, except this would be the small red one and this would be the small blue one. Or this would be the big blue one and this would be the big red one. Okay, so we go through meiosis 2. The chromosomes condense, the chromosomes line up on the metaphase plate. You can see here, this is just random now. It doesn't matter if the blue one's up here or down here. This order doesn't matter because once the sisters separate, okay, the new cell gets both of these in a nucleus that's three-dimensional, so it doesn't really matter what order they're in here. Okay, so prophase 2, the chromosomes condense. Metaphase 2, they line up on the metaphase plate attached to the spindle. Anaphase 2, the sister chromatids separate. Telophase 2, cytokinesis continues, the nucleus reforms on each side, and then later, after telophase 2 is done, we have 4 cells that result from meiosis, and each one of these is completely different from the other. Okay, take a moment again to look at these 4 nuclei. Each one of these nuclei contains a completely distinct genome. Okay. These slides just zoom into those slides we just covered so you can see it in a little bit better detail. Okay, and we've already talked about this, the importance of meiosis. Okay, so it produces haploid cells from diploid cells, and the genetic variation produces cells that are no longer identical to the parental cell. And this variation occurs in 2 ways, by crossing over between non-sister chromatids and by independent assortment of that pair of homologs that happens during anaphase 1. I might have just misspoken that. Let me say that again. The genetic variation occurs by crossing over, which occurs in prophase 1. And the second way is by independent assortment. Okay, so what happens is the homologs line up in metaphase, and then they get separated during anaphase 1, and the way they get separated is random. All right, and then, so we have this haploid egg and this haploid sperm, and when they fertilize, when the sperm fertilizes the egg, we combine those chromosomes, okay, into a single nucleus, and so that brand new genome is completely unique. And offspring are therefore never the same as the parents. And that is the advantage of sexual reproduction. Okay, the long-term genetic variation increases the survival of a species. Why? Because on Earth, the environment gradually changes. Things get warmer, things get cooler, what's in the air changes. Sometimes the oxygen levels go up or they go down, or carbon dioxide goes up or goes down. The amount of water in the air goes up or down. Deserts become lush. Forests become deserts, etc., etc. And these changes that occur lead to extinctions, or they lead to the stress that is the driver behind natural selection. All right, so that concludes meiosis. Now we're going to talk just briefly about comparing meiosis and mitosis. Okay, so DNA replication occurs only once before either of these events, meiosis or mitosis. Okay, so we have S phase. Meiosis requires 2 divisions, whereas mitosis requires only a single division. Meiosis produces 4 daughter cells. Mitosis produces 2. Okay, so if this cell is going through mitosis, it's just going to create 2 genetically identical cells. If a cell is going through meiosis, it's going to create 4 genetically distinct cells. Okay? 4 daughter cells are haploid. The 2 from mitosis are diploid. Okay, so these cells, the parental cell is 2n. The parental cell is always 2n, diploid. In mitosis, the daughter cells are also 2n. Okay, and in meiosis, the daughter cells are n. And the 2n to n happens in meiosis 1, which is called the reductive cell division. Okay, meiosis 2 looks pretty much the same as mitosis. Okay, and then those daughter cells from meiosis are genetically variable, while the ones from mitosis are genetically identical. Okay, so these 2 cells are the same genetically, and these 4 cells are completely different from each other for the reasons we talked about before. Meiosis occurs only at certain times of the life cycle of sexually reproducing organisms. Okay, so basically once you reach reproductive age, that's when meiosis begins in earnest, and you start to produce your sperm if you're male. If you're female, you already produce your eggs when you're actually developing as a fetus. Okay, and mitosis takes place continuously in all tissues as part of normal natural growth and repair. Okay, so this is a side-by-side now between mitosis and meiosis. Okay, and you can see each of these events different. Here's the biggest difference right there. Oops, I didn't really circle that very well, did I? There's metaphase 1 of meiosis versus metaphase of mitosis. And you can see in mitosis, they all line up. All the chromosomes just line up, and the sisters separate. In metaphase 1, the homologs pair up. There's organization here. Okay, this is a thing. This is a thing. And actually, in other classes, we would learn that these things are called tetrads or bivalents, but you don't need to worry about that. Okay, and then you can see telophase occurs, and then I like how in this diagram they basically just squish all of meiosis to here, just to try to make it the same as much as possible to mitosis. All right, so there's your comparison visually. Okay, and these slides just break out those other slides. You can use these to study as well. They're in the PowerPoint. Okay, and then now this is just a wordy PowerPoint slide that tells you everything I just said, basically. So I'm not going to sit here and read this. You can read this on your own. Let me just take a moment here to make sure there's nothing in here that I didn't already talk about. All right, so these are just restatements of the comparisons and contrasts. So the homologs pair and cross over during meiosis, but not mitosis. The paired homologs align at the metaphase plate as opposed to the sister chromatids aligning at the metaphase plate in mitosis. And the homologs separate and move in meiosis 1, whereas the centromeres split and sister chromatids move in mitosis. Okay? And these tables are just good summaries of that stuff as well. Right, so really what I want you to do is not just understand the mechanisms of meiosis and mitosis, but I want you to be able to compare and contrast them, understand them in the context of each other. Okay? Okay, so now that we understand meiosis, let's talk about the human life cycle. So as we've talked about already, both mitosis and meiosis are required. Okay, at fertilization, a haploid sperm and a haploid egg fuse. Right, and so remember this N here, that just means that they have one set each of the complement of chromosomes. And when the egg and the sperm fuse together, they form the zygote. And the zygote is diploid, okay? It's diploid, meaning it's got 2 chromosomes of each unique chromosome. So you would say that there is a pair of chromosomes for each. You would say that there are 2 sets of chromosomes. And then the fetus divides by mitosis for growth and development. Okay, and after birth, mitosis allows continued growth and tissue repair. In male humans, and really in mammals, meiosis in males happens in the testes, and it is called spermatogenesis. And spermatogenesis is a specific type of meiosis that produces mature sperm cells. And in females, that process happens in the ovaries, and it is called oogenesis, right? And this is probably the only word in the English language that begins with 2 O's. And you would say oogenesis. Okay, so here's a visual of the human life cycle, just like you might see in a tree diagram or for a fish or other things that you see in textbooks. Okay, so you'll see the male and the female each produce their sex cells and through meiosis, okay? And so the female produces an egg, the male produces a sperm. You can see that they're each N or haploid. There's haploid, right? And then upon fertilization, you have a zygote, and that zygote then goes through a very complicated program that leads to baby. Okay, and baby is 2N. Really, all the cells once you hit the zygote are 2N. All right, so notice that basically the top of this diagram, the blue area, this is the diploid phase of the human life cycle. And at the bottom here, this beige area, that is the haploid phase of the human life cycle. Most animals that are sexually reproducing, you can divide their life cycles into a haploid and diploid phase. And in, for example, plants, the haploid phase is just as prominent as the diploid phase. This is called the alternation of generations. Okay, so the trees that you see outside, those are actually haploid. And then inside, say, a pine cone, there's a teeny tiny little organism that is diploid, that eventually grows and divides and goes through meiosis and becomes the haploid adult again. Alternation of generations. Okay, I might have misspoke that a little bit. I don't have notes in front of me. I'm reading off the, thinking off the top of my head about trees. So I might have gotten a detail wrong here and there in that last little discussion. All right, so how does spermatogenesis work? Very simply, we're not going to really cover this in too much detail. I'm going to skip this slide and just show you the image here. Okay, so in spermatogenesis, we start with a stem cell. Okay, a stem cell is a cell that, how did we get the stem cell? Well, there was another cell that divided in half. Okay, and this cell here is now going to go through meiosis. This cell is going to perpetuate the generation, so it's going to also split in half, and it's going to make 2 cells. And this cell will go through meiosis, this cell will go through mitosis, and that's how stem cells work. There's always a continuous supply of stem cells, and each generation, one goes and does something, and the other stays as a stem cell. All right, so in this case, this stem cell becomes what's called primary spermatocyte. And the primary spermatocyte, which is a diploid cell, goes through meiosis 1 and becomes secondary spermatocytes. And the secondary spermatocytes then go through meiosis 2, and those become spermatids. And then the spermatids differentiate. They go through a process we can call metamorphosis and maturation, and they become a adult sperm cell that is haploid. Okay, and so in here, this is where the genome is. That's where all the DNA is. This region here is basically jam-packed full of mitochondria. And then we have a flagellum that allows it to swim. Okay, so the flagella is given its energy by all these mitochondria. In oogenesis, this gets a little bit more complicated. Okay, I'm going to skip these and show you the pictures. Yeah, there we go. Okay, so the first thing that happens in oogenesis is you have a primary oocyte. Okay, the primary oocyte, which is this cell right here. Okay, now notice that it is surrounded by other cells. So this is happening in the ovary. Okay, now what happens here is the first meiosis occurs, but notice that there's still one cell. Okay, still one cell. So this is what's distinct about oogenesis. And so instead of two cells, what's really happening here is here's one cell. Okay, here's the second cell. It's called a polar body. Essentially what happens is one of these cells wins and one of these cells loses. The winner takes the loser's essence, all the cytoplasm, all of the organelles, and basically creates this husk of a mini cell. Okay, why? Because the bigger an egg is, the more energy it has to spew out chemical signals to attract a sperm. That's basically the short version of this. Okay, and so a really large egg cell attracts sperm. So notice that this cell here is arrested in meiosis 1. Okay, we're in meiosis 1 here, and all of the eggs in a female human body are arrested in meiosis 1. They're just waiting for the gentleman caller to come along. Okay, so upon fertilization, when the sperm fuses with the egg's membrane and injects its DNA, not unlike a virus, the cell is then induced to go through meiosis 2. During meiosis 2, the same thing happens where you have a polar body form. Okay, so now we have two polar bodies. And what happens is the egg nucleus and the sperm nucleus, let me say that again, the egg nucleus and the sperm nucleus fuse, and we get a 2N nucleus. And so the resulting cell here is called the zygote. And from here, we go through this whole complicated process of differentiation, rather development. Okay, so the rest of these slides here I'm not going to show you. Those are just for your own benefit when you're studying. So we're going to stop here, okay? Feel free to email me any questions you might have, and I will see you next week. Have a good night.